Good morning, Prof. Dr. Jeffrey Rovi Ryan Japning. We are members from Group 8. For this mini project, we visit two sites on the UNIMAS campus, the Real Living Lab, RLL, and the UNIMAS Lake. Our goal was to observe both natural and artificial ecosystems, identify the biotic and abiotic components, explore the limiting factors, and reflect on what we learned through this field experience. Before we begin, let me introduce our team members. I am Azim, the data integrator, and our chairperson is Xin Yi, and our scribe is Priscilla Ampu Anak Magas, and our fact finder is Andrea Clariss Anak Ajak, and finally, our video maker and photographer is Breatheley Dyell Jampang. Together, we collaborated to complete this project and bring you this video presentation. Now, let's talk about the ecosystem we explored. The Real Living Lab showcases both natural and artificial ecosystems. We observed a variety of natural ecosystems, including a tropical rainforest, a flowing river, and a mangrove area. These environments are rich in biodiversity and show complex interactions between species and their surroundings. In addition, we also noticed some artificial ecosystems such as a greenhouse simulating a terrestrial habitat, aquatic mesocombs with water plants and frogs, and aquarium tanks with fish and turtles. At Unimas Lake, we observed an artificial aquatic ecosystem with fewer species but still important ecological function.
Now, let's take a closer look at the biotic and abiotic components we found in each ecosystem. In the rainforest, we observe the bird's net fern, Asplenium nidus, and epiphyte that grows on the trunks and branches of large trees. It uses the tree for support to access better light in the understory and collects moisture and fallen organic matter in its rosette, forming a miniature nutrient cycle. We also found wood ear mushrooms, Auricularia auricula-judae, growing on decaying wood. These fungi are saprophytes, meaning they help decompose organic matter and recycle nutrients into the soil. The spider lily, Hymenocallis littoralis, thrives in moist, sandy areas but cannot grow without sunlight, making it highly dependent on light as an abiotic factor. We observe big-head ants, Pedal species actively feeding on leaves, which illustrate herbivory, a biotic interaction where insects feed on plants. In the mangrove area, the mangrove trees, Rhizopora apiculata, show probe roots and area roots which help support the tree in muddy soil and allow for gas exchange in waterlogged conditions. We also observe several native tree species in the tropical rainforest, including Nyato durian, Palacium gutta, which thrive in rich, humid soil and require sunlight to grow. Pintawa Artocarpus anisopilus is another light-dependent species, shade-tolerant when young but needing more light as it matures. Kayu malam, Diospyros venosa, prefers humid, well-drained forest areas, and its seedling can survive in low light, showing adaptation to understory conditions. Lastly, Geronggang Grotozilum arborescens is a light-demanding species that grows best in open areas or forest gaps, often found in peat swamp or hill forests. Besides, in the artificial aquarium tanks at the Real Living Lab, we observe carp, Opspromnemus gorami, which use a labyrinth organ to breathe air directly from the surface. Cohly fish, Cyprinus carpio, a hardy species with an omnivorous diet and long lifespan, showing its adaptability in captivity. In the aquatic mesoscome, we have found a few four-line tree frogs, aquatic plants, and algae. These species represent a simplified version of natural aquatic ecosystems. At the UNIMAS lake, we spotted milky storks foraging along the lake edges, indicating the lake shore as a wetland habitat. We also saw some aquatic vegetation, though species diversity was relatively low. The abiotic components in Real Living Lab include high temperature, acidic, nutrient-poor soil, high humidity, and in the mangrove area, saline water. These environmental conditions greatly influence species survival and their adaptation. In UNIMAS Lake, abiotic elements include water temperature, light penetration, dissolved oxygen levels, and nutrient availability. This determines the lake's productivity and biodiversity.
Now, let's talk about the limiting factor, which is the environmental condition that restrict growth, reproduction, and survival in each ecosystem. At the Real Living Lab, which include peat swamp forest area, one of the most significant limiting factor is high temperature. Increased temperature accelerates peat decomposition, releasing large amount of carbon dioxide and methane, which contribute to greenhouse gas emission. The heat also leads to increased transpiration and evaporation, drying out the peat surface and making it highly flammable. This raises the risk of peat fires, which can burn underground and release dense smoke. The soil in this area is typically acidic, nutrient poor and thin, which limiting the growth of many plant species. To survive this challenging condition, trees such as those with buttress root adapt by stabilizing themselves and increasing nutrient absorption from the surface. Water and moisture are also critical limiting factor. When peat dries out, it oxidizes and releases even more carbon dioxide. A lack of moisture also causes habitat loss for aquatic and semi-aquatic organisms and leads to tree mortality, especially for large tropical trees adapted to waterlogged environment. Meanwhile, in the UNIMAS lake, high temperature significantly reduce dissolved oxygen levels in the water, putting stress on fish and other aquatic organisms. Nutrient deficiency, particularly of nitrogen and phosphorus, limits the growth of phytoplankton and aquatic plants, which disturb the lake food web. This may lead to oligotrophication, where water appears clear due to low algae growth but support very little life. As water level drop, temperature rises more quickly, further stressing aquatic life. The concentration of pollutants and nutrients increases, possibly leading to algal bloom, which consume oxygen at night and may produce harmful toxins. Low oxygen and high temperatures combined can result in mass fish death, while aquatic plants either dry out or lose root support due to falling water levels. In both ecosystems, these limiting factors directly impact biodiversity, ecological balance, and the sustainability of the habitat.
This field visit helped us connect classroom knowledge with real-world ecosystems, seeing interaction like ants feeding on leaf or fungi decomposing wood made concepts like herbivory, decompositions, competitions, and adaptation much easier to understand. We also improve our observation and critical thinking skills, learning how to distinguish between natural and artificial ecosystems and to identify important biotic and abiotic factors. The greenhouse and aquatic mesocosm show us how artificial ecosystems can support education, conservation, and sustainability research. Most importantly, this experience reminded us of our roles as responsible citizens. We realize that healthy ecosystems provide more than just beauty, they support social well-being, local economics, and long-term survival. Understanding limiting factors also made us more aware of how fragile nature is and how important it is to practice sustainable behaviors.
In conclusion, this mini project gave us meaningful insight into the ecosystem of universe. We observed its biodiversity, unique species adaptation, and the powerful influence of limiting factors on both natural and artificial environments. Through the experience, we learned to appreciate the importance of ecological balance, sustainability, and environmental responsibility. We hope our video encourage everyone to be more aware, more curious, and more committed to protecting the world we live in, starting with the ecosystem around us.